Good evening and welcome to More Songs for Cynical Scientists. I'd like to take this opportunity of telling you what's been happening in the lab since I last addressed you. First slide, please. Now I'm sure you'll all remember what I said here last November when I said what I had discovered with such ease. First slide, please. For the sake of time reduction, I'll miss out the introduction and go straight into the details. Next slide, please. Next slide, please. This shows NMR 3D of MPF and TBP. We PCR'd P53 and I think cyclins A to Z, as you can see. Next slide, please. Can you hear me at the back? Well, can you hear me at the back? Oh, now the microphone's got feedback. Next slide, please. Next slide, please. Please, can someone shut that door or stop the rock group in next door? And please, can someone kindly catch that swarm of bees? Next slide, please. I'm sure you can't make out the tracks, 'cause this has just come in by fax. I'm still expecting several more, so next slide, please. Next slide, please. Now this curve that goes straight down shows stimulation upside down or just a crack the wrong way round. I'm not quite sure. Next slide, please. Can you go 13 slides back? Or is it 16? I lose track. And no, I meant the left projector. Next slide, please. Next slide, please. Now this is looking rather blurred. Is it a plane or just a bird? I should have skipped that 7th whiskey. Next slide, please. Next slide, please. This may cause some slight confusion, 'cause it shows cold nuclear fusion. Now it's melting the projector. Next slide, please. Next slide, please. Have I been drinking too much liquor, or is that smoke there getting thicker? Can you find a new projector? Next slide, please. Next slide, please. It looks as if I'll have to shout. The fire alarm is winning out. Can you just try to pay attention? Next slide, please. Next slide, please. I don't think this slide can be seen. That fireman's hat obscures the screen. Can you just wait until I've finished? Next slide, please. Next slide, please. Now I'm feeling rather manic, but the audience's panic won't disturb my concentration. Next slide, please. Next slide, please. Now this looks like apoptosis. Can you turn off those fire hoses? 'Cause the water level's rising. Next slide, please. Next slide, please. I'd like to thank the organizers, those two penny-pinching misers, who would only guarantee my one-way fare. Last slide, please. I see the fireman's going frantic, so I'd best swim the Atlantic in despair. This is a nightmare experienced by most scientists. As I was walking one summer's morning, I made my way to the library in search of wisdom and education. I scanned the journals so eagerly. I picked up Nature and looked inside it, and lots of adverts I there did see for PCR and restriction enzymes, for cloning kits and HPLC. At last I tracked down the page of contents and scanned the titles so eagerly. There were quarks and quasars and plate tectonics, T-cell receptors and HIV. And as I browsed through the latest scandals, I sighed and yawned rather wearily, till there I saw my latest paper, and brightened up most immediately. I swelled with pride as I gazed upon it. What finer gels could you ever see? And every figure was as we'd planned it. Models of logic and clarity. But then I paused in deep contemplation. A nagging doubt stirred to trouble me. It was just 3 days since we'd sent the paper. There was no way this could be by me. The author's name and address were different. All other features were just the same. I gazed in horror at every figure. There was no doubt we were scooped again. I muttered oaths as I gazed upon it. I mopped the sweat from my fevered head. I'd have torn my hair, but that posed a problem. So I sighed and wished that I'd stayed in bed. I found a keyboard and started writing. A short erratum I sent straightway, correcting names and address of authors. And whistling softly I strolled away. That described one familiar sort of scientific nightmare. This describes another. It's not about reading the literature. Instead, it's about international travel. And in particular, it's about international airports and the stress that these impose on our lives. And for those of a nervous disposition, you'll be pleased to know that at least one airport caters for passengers of a nervous disposition, namely Narita Airport in Tokyo, where the instructions of how to find your way through the airport give a phone number at the bottom of the sheet, which you can dial for fright information. This song is about an airport nearer to home. One perk of being a scientist is that you get to go to exotic destinations like New Jersey and Glasgow. But there's one snag I'll tell you, for no matter where I go, it seems I always have to fly from London Heathrow. Up and away is where I long to go, but why, oh why do I have to fly from
Lucas, I'm still listening to the music. The music is still on. Okay. So, welcome back to the last day of your Zellbiologie practical course. I would like to invite you to go back to the very same question that I introduced to you on day 1 on Monday. And this related to what determines the fate of a cell. And if you remember, I have told you that cells have to decide whether they should grow, survive, differentiate, or die. And in many ways, the concept of these cell phase decisions is all about keeping the balance, all about keeping homeostasis. And in several ways, homeostasis is kept by, let's say, two arms in this balance. On one hand, you have differentiation and death, and on the other side of the balance, you have the rate of survival and proliferation. And should a situation occur, like depicted here, in which one has increased proliferation or increased proliferation is observed, this could have severe effects. An example is shown in the bottom. So let's imagine that you have a normal epithelium. Increased proliferation can precede dysplasia and a formation of a benign adenoma that can then, under certain conditions, develop into a carcinoma in situ. So you see here very clearly a link between increased proliferation and cancer or tumorigenesis. And this link between both has been recognized already a while ago. So you see on this image that is depicted a picture from a review from 2011 that represents the hallmarks of cancer. And one of them, shown in green, is sustained proliferative signaling. So tumor cells require the sustaining of proliferative signals that promote proliferation. Now, cancer and proliferation are, of course, intrinsically linked also to the cell cycle progression. And this is a very quick reminder. Within the cell cycle, we have a phase of DNA replication, or the S phase, and a phase of division, nuclear division and cytoplasmic divisions. And these are flanked by the two gap phases. So, right now, and this is like, say, a warming up stage, I would like to ask you a question. Under certain conditions, the cell can exit the cell cycle, and they can phase in which they are neither preparing to divide nor actively dividing. What am I referring to? This is your so-called warm-up question, and please reply to that via the chat function. I'll be waiting for it. Meanwhile, we go through the topic of today's talk, and that is cell viability and proliferation assays. And these assays are done in order to test the response of cells in culture to different stimuli. These tests can test cytotoxicity or a drug's IC50. Both parameters have been introduced to you already by Taras. And they can also be used, these proliferation assays, in order to detect differences in the proliferation rate. Now, there's many assays that are possible also means that you have to consider what would be the choice of the method. And the choice of the method will in many ways be determined on one hand by the number of cells that you have available to perform the method, by the type of cells that you have available, and also by the expected outcome. And I would add to the list that also an important aspect in the choice of the method is, of course, what kind of equipment you have access to, because the access to equipment will determine in many ways what kind of methods you can use. And these cell proliferation and viability methods are testing or monitoring several parameters. You can analyze the number of cells over time, the number of cellular divisions. You can analyze metabolic activity or take a look at the process of DNA synthesis. All of these can be used to determine cell proliferation and cell proliferation assays. And some of them are directly linked to the proliferation per se, so they are direct methods, and some of them are so-called indirect methods that are linked but not directly measuring proliferation. So, until now, we have no answer to my initial warm-up question, so I really hope that you are there. But in between, and in order to continue with this process, I would like to bring you back to the next topic. And that means, what methods are then available? Well, the available methods that we have in order to study cell proliferation are actually depicted in this picture over here. So you see some of them. This is not an extensive list, but it gives you a broad overview of the diversity of the methods available. The initial and some of the most reliable methods to measure cell proliferation are based on the nucleoside analog incorporation. What this means is shown in the next image. So nucleosides are actually incorporated into the replicating DNA during S phase. So there is a specific phase within the cell cycle in which they are intercalated into the newly synthesized DNA, and scientists use that as a measure to detect a specific phase within the cell cycle. Now, the difference between the different nucleoside analog methods is based on the method that is used in order to determine the rate of incorporation, in order to identify that a nucleoside, in this particular case thymidine, has been incorporated. And you can use radio-labeled thymidine, but you can also detect incorporated forms or analogs of thymidine in different ways. And depending on the approach you use for the detection, you can have different methods. And I now would like to make a short break. I know you didn't reply to my initial question, which was related to the G0 phase or quiescence, but this is an important phase that relates to the cells in which they are basically not dividing, they have exited the cell cycle, but in which the cells still have metabolic activity. And I would like to give you the answer to that question, even though I haven't seen any of you replying to it, simply because at this stage I would like you to introduce you to the next question. And the next question relates to this nucleoside analog incorporation. So, from these nucleoside analogs, I've mentioned that thymidine is the one that is used in the studies of proliferation. Do you have any idea why we look at thymidine incorporation and not one of the other nucleosides? What would be the reason behind it? So this is a tough question, but I'm sure that you can manage it. Try to consider what would be the difference between thymidine and the other nucleosides. And meanwhile, we'll go back into some examples of this nucleoside analog incorporation. And an example coming from our team is depicted here. So what we see here are frozen skin sections from mice, so taken from mice, that have been drinking water with BRDU. So the BRDU can be used for in vivo labeling by adding it simply to the water. So you see that the mice take up the BRDU and they label the BRDU positive cells within their tissues, and you see that here on cytochemistry on frozen tissues. So you see an epidermis. This is an epidermis section in which the white line corresponds to the basal layer. This is the basal layer of the epidermis. And you see that the BRDU is incorporated in some of the cells that are within the basal layer of the epidermis, so the bottommost layers. And just as a reminder, I hope you know that already, but the epidermis is formed by proliferation of the cells on the basal layer that then differentiate, and also during this differentiation process lose their nuclei. That's why you see here the nuclei standing just on the cells adjacent to the basal layer, and then you see that on the upper layers the nuclei are lost. The limit of the epidermis is in this staining not seen. But you can use this approach. Oh, I see an answer from Helena. Thank you, Helena, for letting me know that there was a lag on the stream. Unfortunately, at the moment, Professor Huber is not here, so I hope that this issue with the lagging will become better. In any case, Isabel already replied, and this is absolutely true. You use thymidine so that we can discriminate between the rates of DNA synthesis and the rate of RNA synthesis, and that's the point. So because you with thymidine, you have done during the RNA replication uracil, so you can discriminate between DNA replication and RNA replication. Thank you so much. Thank you, Isabel. And we come back to the topic that I was referring to. So you see here an example of staining of both sections from both mice. In this case, we have a control mice and a knockout mice, and you see that the number of the BRDU positive cells within this section is different. And therefore you can use this snapshot into the tissue of the mouse and simply count the number of cells per view, so per view field. So this nucleoside analog incorporation is an effective method. As I mentioned to you, is one of the most reliable methods to measure proliferation, and it is very versatile. So you can combine it to the detection in immunohistochemistry and immunocytochemistry, but you can use it also in combination with FACS analysis, and you can also do ELISA, for example, on samples that have been treated with BRDU. So it's very versatile. I would say that one of its major disadvantages is the fact that you're actually just taking a look at the snapshot. Like I mentioned, in this slide you see the number of cells that are undergoing cell division, but you do not have a notion of the rate, so you don't have a process over time that you're actually analyzing. And this I would consider the major drawback of these methods. So, having said that, this gives you a summary of this nucleoside incorporation method, and I would now like to change into the next group. And the next group are the so-called cell cycle associated proteins. And the cell cycle associated proteins include, as it's stated here, Ki67. So there are several of these cell cycle associated proteins that can be used as markers for proliferation. I am showing here a few which of the most commonly used ones. And as you see, they can be used and integrated in through different methods, either detection by flow cytometry, FACS, or by histochemistry, but you can also, some of these markers, for example, use them on Western blots. If you ever want to detect proliferation in the Western blots, that's another issue. But they are very reliable markers. And interestingly is that most of them associate to one or the other phase within the cell cycle. For example, the phosphorylated form of histone H3 is integrated during M phase, so it peaks during the M phase of the cell cycle. And you see also here on the list Ki67 that was annotated in the table that I've shown you before. Ki67. Do you have any idea why Ki67 has become such a popular model, such a popular protein in order to detect proliferation? I know this is a hard question, but I hope that you will have a feeling for it. So I can tell you up front that Ki67 has been identified as an antigen in Hodgkin lymphomas, and that this led then to a very prominent use of the specific cell cycle marker in the determination of both the staging, so what stage of the cancer we are referring to, but also it's used in multiple types of tumors. So having said that, try to think about it. What makes Ki67 different from the listed here other proliferation cell cycle associated proteins? Having said that, I come back again to another example, and this comes from the same publication. We look again at the section of the skin of the mice. And in this particular staining, you see here keratin 14 that stains the basal layer, so the stratum basale of this epidermis section. And he also recognized the presence of the phosphorylated histone H3 on cells that are flanking either at the epidermis or at the dermis, but cells that are in the proximity of this basal layer. They are basically absent in the upper layers of the epidermis. Now you might wonder, okay, I see here a tremendous staining. What is this? This is actually a background staining that occurs when you stain for phosphorylated histone H3, and this shows us the stratum corneum. So this is actually not the staining you should be looking at, but tells you how thick your epidermis actually is. And you might wonder why we don't see this line, even though it's background, on the other section, on the other epidermis. And the reason for it is that in this particular case, the mice show a differentiation defect, so they cannot form a proper epidermis. They have a defect in the formation of this barrier, and their very thin epidermis does not contain a stratus corneum. So you don't have this background signal. Important for us in the context of this presentation is that, again, like I showed you before for BRDU, you can also, in the case of histone H3, count the number of nuclei that are histone H3 positive, and thereby estimate the rate of proliferation, or the rate of the cells that are undergoing proliferation within the section that you are observing. Good. That brings me back. Very good. We have, I just took a second because I'm looking at the live chat. So Yasmin says, because Ki67 can be found in every proliferation stage, but not in the G0 phase. Exactly. Very good, Yasmin. This is exactly the point. So because it can be found in every stages within the cell cycle, but it's significantly reduced in G0, we can actively discriminate between dividing and non-dividing cells, so cells that have exited the cell cycle. And this is important because, for example, in the terms of histone H3 that you see here, we are only detecting cells that are under mitosis. That means cells that are, for example, in G1 or G2 or the S phase, we are not seeing them. That does not necessarily mean that they are not, for example, preparing to divide on an S phase, but just that the type of staining that we are using doesn't allow us to identify them. That's one of the reasons why Ki67, and Yasmin pointed exactly to the point, has become such a popular tool. Good. Now that we discussed about the cell cycle associated proteins, I also would like to stress that, like in the example before with BRDU, this gives you a number of the proliferating cells. So, how many cells are at that stage undergoing a specific phase of the cell cycle, and does not tell you the rate of proliferation. So we have no notion over time. We don't have any prolonged time frame readout. Good. The next group of viability and proliferation assays are the so-called cytoplasmic dyes. An example of them is annotated here, and this is CFSE. What are those dyes? These are cell-permeable dyes, so they can actively pass through the plasma membrane, and they then associate with amines present in proteins that are found intracellular within the cells. And this staining is very robust, it's stable, and you can detect it both live or you can, via aldehydes, fix the signal. The advantage of such proliferation dyes is that they can be used in the kind of pulse chase experiment. So you can use them to label an initial population of cells and then see what happens over time. The picture that explains this is shown here. So you see that we have labeled the original population of cells. And when the cells undergo cytokinesis, the content in terms of dye is split among the daughter cells, so that the daughter cells, each one of them, has a half of the content of the dye that is found in the mother cell. That allows us to look at so-called generations. So you can see in the population of cells that you have in a plate, what are the amount of, let's call it mother cells, and what's the amount of cells that are on the second, on the third, or on the fourth division afterwards. So these approaches are dependent on the detection of fluorescence. And you can imagine that it could be suitable for use together with a microscope. But the fact is that most of our microscopy systems are actually not very quantitative. They are just qualitative. And that poses an issue, because in this particular case, the measurement should be as quantitative as possible. And that's the reason why, in particular, in the case of this called cytoplasmic proliferation dyes, they are associated with the use of flow cytometry, FACS, because we need that quantitative information. Now, you see on the list, CFSE that was shown in the previous table is, let's call it the father dye, the first one to be initially identified, and it's a green dye. But there are many others. Shown here is, for example, trace violet, which was the second one to be found. But if you look and Google into most of our commercial companies nowadays, they can provide you dyes nearly throughout the entire spectrum, so all colors of the rainbow. That might depend actually on your needs, on what you want to stain or co-stain the cells for. Good. So we are through with the cytoplasmic proliferation dyes, and that brings me to the next group of assays to measure proliferation. And this group is very broad, very heterogeneous, and comprises all of those methods that are indirect, so that do not measure proliferation directly. A subgroup of these indirect methods are those that involve labeling or detection of the metabolic activity. And they have been introduced to you already, because Taras, if you remember, introduced you yesterday to the WST1 assay, which is an example of such a metabolic activity measurement. At this stage, and from my side, I just would like to stress again that what you are measuring through one of those assays is the metabolic activity. Of course, the metabolic activity is related to the number of cells, and you can see increases in the metabolic activity that are due to cell proliferation, but you can also detect changes in metabolic activity, even increased metabolic activity, that does not occur because the cell divides, but simply because they change their metabolism. And this is a very important point. So this is an indirect group of methods to measure cell proliferation. And the second big group of indirect methods is cell counting. Our topic of today, in broad terms, counting cells. And usually this counting of the cells is combined together with viability assays. The reason for it is very simple. You can see an increase in the number of cells that is dependent on an increased proliferation, but the increase in numbers can also occur because the cells show a decreased death, so a reduction in cell death. Therefore, traditionally, people combine counting of cells with a measurement of cell viability. And this combination of assays has been introduced to you already by Ilya Vietov on day one. If you remember, and this is also shown here on the bottom of the table, Ilya introduced to you the usage of trypan blue in combination with a counting machine called Luna. So again, a combination between the use of a system that allows you to count cells with the measurement of cell viability. Good. We have gone through, let's say in broad terms, this table and the type of methods that can be used to study proliferation. And now I would like, in short, to bring you back to the bench. Imagine that this is your working place and that you're counting. You're counting, as we've mentioned, we are studying proliferation, the number of the green dots. And as a task, within the next 30 seconds, I would like for you to try and give me an estimate, plus minus, of the number of green dots that you see in this picture. I'll give you time for this question, so think about it and give me an estimation. How many green dots do you see in this proliferation movie? I will play it again, just to show you one more time. I'll wait a little bit until I give you time to count. But remember, the whole idea is to make an estimation. It should not take you longer than half a minute. Give it a guess. How many cells do we see here? How many green dots do we see here? Okay. Isabel says 130 dots, or cells, or green dots. Elias says 120. Do we have another guess? Anyone wants to try? Oh, Elena says around 150. Okay, I have to tell you. I actually took the time to count every single one of these dots. It took me between 4 and 5 minutes on a fixed image, not on a movie. And I can tell you that there are exactly 156 dots. That means that Elena was pretty close to the truth. This image, of course, it's a little bit of a gag joke, but it's intended to bring you back to a point that is very important. And that is that in juggling around which one of the methods to use, we have to keep in mind two parameters. One is time, and the other one is accuracy. That means, how much time do I actually have in order to count my cells? Can I stay here and take half an hour to count them? And on the other hand, I have to take into consideration the accuracy. Is it really important for me to know exactly the number, or can I estimate, like you did, within 30 seconds and say, well, it's a bit more than 100 dots? All of this depends on what your downstream intention is. Am I just assessing that this is an 80% confluent plate, or do I need to know exactly how many cells I have? Well, this assessment and the time that you devote to it is, of course, also dependent on whether you are counting the cells manually, or if you have an instrument or a computer that is aiding you or helping you in the process. I hope this point is clear. So there is no one method takes it all. It all depends on what you want to do and what is your purpose. Now, having said that, we now really go back to your experimental setup. And this is the experiment as we have designed it, and that is included in your protocol book. So, you start this experiment of cell counting with 3 plates. In each one of those plates, we have 3 wells which have cells. And they have been plated at 8 times 10 to the 4th cells per well. Now, in the beginning of their experimental procedure, you have to change the medium that is present in 2 of these plates in order to add 2 inhibitors. Inhibitors and what they do to cells I've introduced to you in the first day of your practical course. So you add to 2 of the plates either Taxol or Velkit in 2 of these wells. And the 3rd plate, you take the content of the plate and the cells in these plates for counting. This procedure is repeated on day 2 and on day 3. And each one of these days, you take one of the available plates in your cell counting chamber, so in the incubator, and count 1, 2, 3, whatever cells are left within the wells of your dish. So let's go back to the protocol. And let me remind you, this is all within the protocol book that you have gotten from us, and it's available through Elias. So if you go to page 33 of this protocol book, what you find is the beginning of this cell proliferation and cell counting experiments. You see that you need these 3 6-well dishes. I've introduced you to them before, and they are represented here on the bottom. You need a stock solution of Taxol and Velcade, the 2 inhibitors. And of course, you need a bit of equipment. So you need Falcon tubes, pipettes, EPIs, and also 2 pieces, very critical for this counting. One is the cell counter, a machine that I will introduce to you later on, and also a counting chamber. And on day 1, one of the first tasks is usually to label the plate. So you need to label the plate in order to know which plate will be used in which day and what kind of treatment will go in each one of the wells within those plates. And once you've done that, we go to the next stage. And that means we need to prepare the solutions of the inhibitors. So we need to calculate the volume, or you will need to calculate the volume of Velcade necessary to prepare the solution that you will be using with the adequate concentration. And you prepare the solution in a 15 milliliter Falcon. And then you do exactly the same thing and prepare the solution at the right concentration of Taxol. And once you have both, you can then aspirate the medium that is present on the respective wells and add the inhibitors into the cells. This means, after you've done that the plates of day 2 and day 3 are actually ready to be placed back into the incubator. Let's imagine that you have done that. The next thing that you still have to do, and this still on day 1, is to count the cells in the remaining plate. So the cells on the wells of the plate that is still left. How to do that? Well, first you have to label EPIs. So you have to label the EPIs 1, 2, 3, so that each one of them corresponds to one of the wells of the plate that you have. Then you need to aspirate the medium from each one of the wells, and then rinse the cell layer with 2 milliliters of PBS. So you have the PBS, and you just add PBS to the cells. Now, I have a question for you. I've introduced to you already on the first day the effects that we expect from both Taxol and Velcade. And both of them, if you remember, leads to an increase in cell death. So, there's an issue here. What do you think is going to happen when you wash your plate with PBS? And I'll give you with that question until we proceed, and we'll continue talking about this method. So think about it. What happens to the cells that are dying when I'm adding PBS? Good. So I've added the PBS, and upon adding the PBS, I then remove the PBS and add trypsin. And in this particular case of this protocol, we are adding a 10x trypsin. So this is a concentrated trypsin. It's different from what you've heard on day 1 from Ilya. And I will explain you on the next slide why. But bear with me. So we add the 10x trypsin. We add 1 milliliter of the 10x trypsin to the solution in the center of the well. And then we immediately aspirate the trypsin. We remove it. Some cells, and this includes also the HeLa, will be difficult to detach, especially because you removed most of the trypsin, and you might increase the detachment by incubating them at 37. So that means that over time, you have to pay attention to the procedure and look that the cells are actually detaching. Good. If the cells have detached, the next thing you would need to do is to really facilitate this process by often just giving a kick into the plate. Just make sure that you see the movement of the cells in whatever little bit of trypsin that is still left. And once you have done that, You can add the medium onto it. You add medium to these cells, and you have basically, by performing this and resuspending the medium, the cells back into the apples. So this is in short terms a process that we call dry trypsinization. And now I would like to take a minute because, yes, Elena already replied, the cells that die are washed away. This is important. Yes, it is, because it means that using this approach that I'm introducing to you, you will always have an underestimation. If you detect any cells that are dying, the number of cells that you detect as being dead cells will always be an underestimation of the real number of dead cells, because those that are dead will be washed away during the PBS wash. That means that the most reliable number that you will obtain is actually the number of the cells that are alive. So please bear that in mind when you analyze your data. Exactly, Madalena, Lucas just informed me also via the chat. Magdalena also stated that it can further remove dead cells from the plate. Yes, and it's exactly this removal that means that the number of dead cells that you will detect in the end of the procedure is going to be an estimation, an underestimation of the real value. So the most reliable number that you have is always the number of the cells that are alive, those that are actively kept because they are bound to the plate. Good. I would like now to take a minute and bring you back to this story that I've introduced you to a method that is different from the one that Ilya introduced you on day one. And I would like to explain you why we use then different changes in this protocol. So in the first day with Ilya, you were introduced to a process in which you start by washing the cells in PBS. This is common to both methods. Then you add 1 times trypsin, you add it to the plate, you incubate, you then add medium into the cells and put them on a falcon. And this falcon with cells in medium is then centrifuged. And upon centrifugation, you resuspend the pellet in fresh medium. And this is then what you use to count the cells. Now, what you've heard from me in the past slides is actually that under certain conditions, and this relates normally just to counting, it might be of an advantage to use the 10x trypsin, immediately remove the trypsin, and incubate these cells with just a very little amount of 10x, so highly concentrated trypsin, and immediately afterwards add the medium. Why do we then use this, let's say, in many ways simplified protocol for counting? The reason behind it is because the centrifugation step and the resuspension that you have to perform afterwards can strongly influence the number of cells that will be viable afterwards. So this means that by resuspending and pipetting up and down, you can increase the rate of cell death. Now, if you're cultivating the cells, the approach that Ilya introduced to you is the right one. If there is any additional cell death, this can be easily neglected. This is not the main topic. And this is, so to say, the canonical method of cultivation. Now, in a circumstance in which our main objective is to count the cells, and we expect increased cell death, which is the fact in this particular case, because we are using Taxol and Velcade, it can be an advantage to avoid this centrifugation and resuspension method because it will interfere with the results. And it could be that this additional increase in cell death, so a reduction of the viability, might mask some of the effects that you would like to see downstream, the effect of the inhibitors. So this is the reason why we have 2 different methods, and it all depends on what you want to see, why you want to use them, what's your purpose. Having said that, let's go back to cell counting. You see here the 2 pieces of equipment that I would like to introduce to you in this practical course. One of them is a counting chamber, the second one is the CASY counter. So let's start with the counting chamber. Imagine that we start in the step I left you before, so we have cells in suspension and we took an aliquot of it on an epi. And now we would like to count the cells. So these counting chambers contain two different counting areas. I'm pointing to them now. I hope that you can see them. These are these rectangles over here. And these counting areas are flanked by two holes, left and right, that are also shown here in the lateral view. And the counting chamber also uses a glass. So like a cover slip. They are a little bit thicker than the cover slips that you use for immunofluorescence, but they are used to place on top of the chamber. And it's the combination of the, let's say, width and length of these counting areas and the distance to the glass that identifies a volume. And this volume is important because it will allow us then to calculate back how many cells do you have in solution. So you see, and this is not per chance, that always these counting chambers have a reference of, you see here the area, and also Tiefe, so the depth. So these two parameters will allow you to then calculate the number of cells. So we start by actually adding, pipetting the cell suspension, into this counting chamber. And if the volume of this solution is not exactly the same as the amount that would fit into this volume, it's not a big problem, because you have here these two lateral spaces, left and right, through which any excess of the solution can go into. So there's basically a method in order to avoid that this area is overflowed. And once we have done that and correctly set up the amount of the volume of the cells that are in this area, we can then use this counting chamber and place it under the microscope, and then simply count. So, having said that, I'm bringing back to the counting chamber. So this is a video that I've prepared, in which I show you that you actually grab the plate from the incubator. You've seen me and others doing that before. And then you go and cross-check that the plate is labeled as it should be. So you see here, this would be a plate of day 2, the second day, and we have the untreated or control cells, plus the cells treated with Taxol and Velkit. And immediately adjacent to this, so once you have recognized that this is what you would like to actually trypsinize, resuspend, and count, you can start with a procedure. Now, you have been introduced to this splitting protocol before. As I've mentioned, it's the same protocol you have seen from Ilya on day 1. Therefore, I skipped it in this movie. And you should bear in mind that for counting the cells, this protocol has to be optimized or has to be changed with these little changes that I've mentioned before and that are critical for a dry trypsinization. So you use the modified version, the dry trypsinization method, and you then obtain basically samples within 3 EPIs. So I would like to start then from this step. So you have the 3 EPIs labeled and already containing the solutions of cells in a medium, and then you take them up and bring them into your counting chamber. And the first task is actually to confirm that the counting, so sorry, I'll go back for a little bit, just to confirm that the glass is actually standing properly on top of this chamber. Bring it back, move it for a little bit. You see that I've confirmed that the glass is properly fitting, and then and only then do I add the solution of the medium containing the cells into each one of those counting areas. One here and one there. So you see I put the pipette diagonally. This is one reason, because I have to fit it in here, in between the glass and the counting area. And you fit one area, so one of the counting squares, and you fit the second one. Good. Once that is done, you put the rest of the sample back, and you can transfer the counting chamber into the microscope. This allows you then to zoom in and select which area would you actually like to count. And I will stop the movie now to show you that what we look under the microscope is this little cross. You see the cross here with very tight lines adjacent to each other. And you see that in each one of the surrounding areas you have also lines, but they are spaced. They are more spaced. There is more space between them. And this is very important and will be important for which areas of this counting chamber should you actually count. So as you see, I'm actively moving the chamber in order to recognize one of the side quadrants, so one of the side squares that is adjacent to the cross. And this is the end of the movie. So it's the definition of the area that you should count. So let's go back to the counting. I've mentioned to you that we have 2 counting areas. Each one of them has a cross. And what you saw on the movie is that actually we've selected the bottom left corner, so this quadrant in the bottom left corner that is visualized here. So this is the area that you just saw in the previous movie. So what do we actually count? For the practical course, we decided that you should count 4 quadrants, so the 4 areas that are on the corners of the cross. And what you do is that you count the number of cells in each one of those squares, and you count all of the cells that are within this big square and also those that are on the left and bottom line. You don't count the top or the right line. And the reason for it is because under certain conditions and in certain cases, it could be important to count more than those 4 quadrants. And therefore, people have decided that the easiest way would be to avoid 2 sides so that you avoid counting the same cell twice. So for example, counting the same cell in this quadrant and on the following one. So in order to avoid that, you also always count those that are just on the left side and on the bottom. And very important is also that you count just the cells. So don't count things that are smaller than the cells. For example, this is a cell. You can see it is a round sphere. You can see this halo around it. Don't count this. This is a little piece of dirt. I hope that you can recognize where the cursor is pointing to. This is not a cell. It's much smaller. And in the same way, you should also not count if you see agglomerates. Yeah? So just count cells. Or if you see, and sometimes it does happen, a hair follicle. Don't count hair. So it's just restricted to cells. It sounds basic, but sometimes we have the students being a bit puzzled, especially in the situations that there is no cell visible. And this can actually happen. Remember that you're treating the cells with inhibitors that cause cell death. So maybe on the 3rd day you will see one or the other quadrants that has no cells. And if you see no cells, then you count to 0. That's no problem. That's how the system works. So be correct. And the idea behind it is you count the cells on each one of these quadrants. You divide it by 4, you multiply it by 10,000, and there you go. You have the number of cells per milliliter of solution. Good. And with this, I'm nearly over into this first part of today's session, and I would like to give you a small homework. So, the first question is, which of the methods from the introduction table, this table that we have went through, would you use to count? A, T cells isolated from the spleen of a mouse, and B, proliferating cells on a skin biopsy. Please justify your answer. And the second question is, the Neubauer counting chamber has also a different name that reflects its historical roots. Do you know anything about it? Good. So these are the questions related to this first part. This will be part of your homework. And I would like now to make a 5 minutes break. Please remember that immediately adjacent to this session, so today's session, you will also have an introduction from Lucas in what concerns the overall view of your homework and what is intended from you. So please stay in, and I will see you in exact 5 minutes. See you, bye.
Amsterdam, and I park in the long-term bike sheds with no sign of traffic jams. At Amsterdam it's easy as I climb the moving stair and arrive at my connecting gate with loads of time to spare. Up and away is where I long to go, but why, oh why did I ever fly from London Heathrow? So I sit back and relax as my flight takes off on time, when I realize I've left my watch still set on English time. So I slowly realize that although the gate was right, because my watch was 1 hour slow, this can't be the right flight. So if this is not the 10:05, I wonder with alarm if this flight is for Bolivia or Dar es Salaam. So I listen very carefully, but the pilots don't say much, and everything they say in fluent double Dutch. Up and away is where I long to go, but why, oh why did I try to bypass London Heathrow? So I listen very carefully for any English version, as I wonder if I'm going to need Swahili, Greek or Persian. Then the pilot reassuringly suggests we'd like to know that very soon we're due to land in London Heathrow. Up and away is where I long to go, but why, oh why do I have to fly from London Heathrow? This song is an awful warning. Lord Nelson's renowned for Trafalgar. An apple helped Newton to fame. Ed Southern's great worth spread all over the earth because of the blot on his name. And sometimes I feel green with envy of those who reach fortune and fame. But if fame knocks at the door, then I won't be so sure. If you listen, I'll try to explain. For no matter what I do, I fear I'm destined to be known as the man who coined that ghastly term, molecular chaperone. One day I found a new protein that acts in an unusual way. It helps other structures assemble by keeping their ligands at bay. It doesn't bring partners together, but rather it holds them apart to protect their virginity at any salinity by playing a chaperone's part. This protein I called nucleoplasmin, and I wanted to say how it acts, and molecular chaperone seemed a good term to accommodate most of the facts. For this term I had planned just a quick one-night stand, then I'd carelessly throw it away. But I ought to have known that no true chaperone could allow me to act in this way. Now, no matter what I do, I fear I'm certain to be known as the man who coined that ghastly term, molecular chaperone. I'd failed to allow for John Ellis, a true British bulldog of old. With a shotgun he said, "To this term you'll be wed," and he welcomed it into the fold. I never thought that one cute concept could cause me such anguish and pain. But to increase my groaning, he coined chaperonin and used it again and again. So no matter what I do, I fear I'm certain to be known as the man who coined that ghastly term, molecular chaperone. How can I escape from this problem and banish this term from your heads? I need a distraction to divert your minds and leave a new image instead. So I need a new image to help me escape. So I might have to take up fraud, arson, or rape, or even descend to recording this tape to silence those desperate groans. I'm divorcing those damn chaperones. This is politically and ecologically correct. It consists almost entirely of recycled garbage. Namely, some of my previous songs.
So, welcome back to this second part of today's session. And the second part relates to the CASY counter. A quick reminder. We are within this last experiment and we are counting cells. And one of the pieces of equipment that I am introducing to you is this CASY counter. So you see that the CASY counter has system here on the left side that includes some reservoirs. And it's connected normally with an electronic display. Now these reservoirs are different and they fulfill different functions. So the first one is this CASY solution reservoir that is mounted here. Then we have a second one in which you will put your samples in, and you have a waste bottle. And why am I referring to these three reservoirs in particular? Because you have to bear them all in mind when you are using the CASY. And in a lot of ways they define also the trajectory of the buffer and the solutions within counting. And these are defined by the two terms, purge and counting. So if you're purging, the buffer is removed from this bottle here, the bottle containing the CASY solution. It passes through all of the different tubings within the machine, and it's then disposed in the waste. If you press counting, then what you are doing is actually removing sample from the sample reservoir that partially passes into some, but not all, of the tubings within the system and is then pushed back into the waste. So this is just a quick reminder that in counting and purge, you actually use or remove sample from two different positions. One is from the CASY solution reservoir and the other one is from the sample. Now, having said that, I would like to provide you a bit of an overview on how the CASY actually works. And the CASY counter is based on the following. We have a reservoir. This is here on the scheme, but it's the same reservoir that you see on the left side. And this reservoir is filled with an isotonic solution, the CASY solution, which contains electrolytes. And within this electrolyte isotonic solution, you dilute your sample. So there are cells floating in here. And the principle of the CASY is actually that you push the cells through a measuring capillary. And this measuring capillary is flanked here on the bottom by a so-called measuring pore. This measuring pore is very important. And it flanks the measuring capillary. This capillary and the pore are within a so-called artificial precious stone, so it's like an artificial stone in which the capillary is in, and that it also includes an electrode, an inner electrode, that is connected also to an outer electrode, an electrode that is outside of this measuring capillary. Now, when the cell is passing through this measuring pore, it dislodges, so it displaces a certain volume of CASY solution. And this displacement is measured as an electric pulse. So you see a change in electric content. And this change in the electric pulse is in terms of the width and also of the amplitude of the electric pulse directly proportional to the volume of the cell. So let me explain you. So what you see basically is you have a flow of electric current, and whenever you have a cell, an intact cell, a viable cell, the plasma membrane will isolate a certain portion of the current. So you will see an increase in resistance that is proportional to the volume of the cell. If the cell has died, then this effect of the plasma membrane is no longer occurring, and you see a small change of resistance, much smaller than with an intact cell, that is now equivalent to the volume, so to the size of the nucleus. So through this principle, the CASY is able, without any staining, to distinguish between cells that are viable or alive and dead cells, because basically it distinguishes between the volume of an original cell and the volume of the nucleus. Good. So this is an introduction to this CASY counter. I've shown you that it uses different parameters to this, the change in resistance in order to determine if the cell is dead or alive. And I have a question. I hope you're still there and active. And the question is the following. So I can use this system to count mammalian cells, all different kinds of mammalian cells. And I'm telling you up front that you can also use it, for example, to count yeast cells, just as a fact. So if I'm counting yeast cells, what in this system would I need to change, if anything at all? So I will leave you with that question, and I would like to come back to your protocol book. So again, we are using the protocol book that you have obtained, and we go back to the part of the protocol book that refers to the CASY counter. So we start by turning on the CASY. Then we confirm that the correct capillary is mounted, and if necessary, change it. Remember, this measuring capillary is the one in which the cells will pass through, and this passing of the cells, the counting, will occur at this measuring pole. So then you need to confirm if you have all of the tools that you need. So do you have enough reservoirs? Yes or no? And remember that you need the reservoirs not only to count the samples, but also, for example, for blanking. That means in between the samples you need to exchange into a bit of CASY solution that does not contain any cells. And then you need to fill in one reservoir with 10 milliliters, one of those reservoirs with 10 milliliters of the CASY counter solution. This we have done, and insert it into the machine. Then we purge. I've introduced it to you already. This means the CASY solution is passing through all of the tubings within the system. And then, once it has purged, you can select the right program from the menu and perform a blank run. Blank run in this particular case means that we use or take the sample out from the 10 milliliter CASY counter solution, like if it would have been the solution containing the cells, but now without cells, so that we can estimate the background, the amount of background that the machine is giving us. And we have assigned that if this background count is higher than 50, you need to repeat the system, just to make sure that there are no debris, no particles left behind that can interfere with the counting. So now I'm shortly looking at the live chat, and there is an answer from Isabel that says, I guess you need to change the pore size. Isabel, you're actually correct and spot on. because the size of this measuring pore is going to be very important. If you have a pore size that is too broad for your yeast cells, then there is the chance that more than one cell will pass at the same time. And therefore, the counting that is done by this change of resistance will not reflect the number of cells, the number in this case of yeast cells, but it will be counted as one even though you have more than one. Exactly for this reason, we changed the capillary and in particular the pore in order to adjust to the size of the particle, or in this case of the cells that you are looking at. Thank you, Isabel. Let's proceed. So we are in a system that we have blanked, so the background is okay. What do we do next? The next step is actually to add your sample. So you need to add 100 microliters of the cell suspension, in this case the untreated sample, to the counting tube, to the tube you already had, that contains the 10 milliliter of the CASY solution. These volumes are really important, so you always have to add 10 milliliter of the solution and 100 microliters of the cell suspension. We'll discuss why in a few minutes. Then you close the reservoir, flip up and down so that you nicely resuspend it all, and insert the reservoir into the CASY. You will see how that happens in a movie in a second. And then you let it run. So press the menu and let it run. The only thing that you have to do now is to look at the results that are displayed on the machine and then annotate, so make notes of what's the number of the viable cells, death cells, and the increased viability. So you generate a kind of a table with those values. Then you fill a fresh reservoir with CASY counter solution, put it in the machine, and select purge. You're again removing everything that might be left from the previous counting. And you repeat the procedure, like is stated here on step 5, for all of the remaining samples. So in this case, with Velkit or Taxol. So you just repeat the previous steps. So once you're done and you have counted the untreated, the Taxol, and the Velkit treated, you have to more or less bring the machine into a state that it can be used by your colleagues. So you always have to end up by, on one hand, purging, meaning making sure that all of the buffer in the solutions contains no cells or cellular debris, and you have to perform a blank to make sure that everything, even the volume that is taken up from the sample and it's introduced, so the tubings that is introduced into the CASY counter, is also free of cells. Once you have done that, you can then turn the program off. So you now got a bit of an overview how this machine works. It's actually really, really simple, and would like to demonstrate how this is done. So you see me, and I am actually selecting and mounting the capillary. So I realized that the capillary that had been mounted by my previous colleagues had been one that is used for Saccharomyces cerevisiae, and therefore I'm now exchanging it into a bigger capillary. You see that the capillaries have numbers here, labeled on the top, and there is another one now that I'm just actively taking up, which is the right one for mammalian cells. And you now have to put the capillary in. You see that there is a little tube in here, and you have to screw. You have to actively turn around and screw the capillary into screwing cap that is on the top. So this takes always a little bit. You have to assure that it's stable and stably inserted. Round, 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 and it's now fixed. And after you've done that, you just close the reservoir of the remaining capillary that always should contain solution. So you see that there's solution in here. And then you just fill a reservoir with 10 milliliter of CASY solution and put it in. Now notice that while putting, you have to make sure that you introduced into the system also the external electrode. If you don't have both electrodes, the system will not work. So pay attention, let's stop this for 1 second, that you have the outside electrode also within the reservoir, okay? So now that everything is done, you can purge the system. So you just tell the machine to go into the display and purge. And the purging takes place. Once you've finished with the purging, you then have to blank. So this was the next step. So we already have a fresh reservoir in here. So we can select the program, the adequate program. In here, we select the program for the HeLa cells, and we go to measure. So this will pass the liquid through the entire system. So you see that the machine is progressing, and you can actually observe in the CASY that it's taken up from the sample, because you see here the displacement of the liquid in the reservoir. In a few seconds you will see that there are some light coming up and you see liquid flowing. I hope that we'll see it again. You see the little bars and liquid flowing. Good. So you also should control that whatever liquid is taken up, it's also disposed in the waste, and make sure that the waste bottle is not full. So all of these things you should do before you actually start and load your sample. So it's the pipeline and all of the tubings is correct, and we see that the sample is being taken and deposited in the waste. I hope you saw that on the previous movie. So now that you're done, we can go fill in the 10 milliliters and load the sample. So you see me grabbing a pipette, the sample, shaking the sample to make sure that you have an homogeneous suspension of the cells, and then loading it into the tube. Now you shake the tube, again making sure that it's completely resuspended in homogeneous solution, and then we turn it on. So pay attention. I hope that you have seen that the tube is kept in place by this pedestal that you have to turn around in order to assure that it doesn't fall off. So it's placed in, it's turned around so that there's a bottom onto it, and then the sample is loaded. So now you can go in and say, yes, I want to measure. And the measuring process starts. It always takes a few minutes. Once this is done, you can observe the results. And I would like to stop now to show you two things. The first thing is, because of the principle that I've introduced to you before, the CASY assigns limiters in diameter that define what is a dead cell and what is a cell that is alive. And these distinctions are shown here in these little numbers. You will see a zoom in a second. That defines, let's say, the micrometers that define these are dead cells, these within this measurement, and that one corresponds to the range of viable cells. This is very important for you because it reports to the overall distribution of all of the counts within a general view. So if we zoom now to the data observed, you can see that we don't only have this graphical representation with the numbers. You can now really see them. So we have here the 12 micrometers that defines the definition between what we call death and what we call viable cells, and this you can define for each cell line individually, so you always have a different program for each one of the cell lines you use. And you can see that the CASY has calculated both the number of dead cells, but also the number of viable cells, and everything that is smaller than a dead cells, it counts as a debris. Now, based on this, I can tell you, and on the data from the CASY, that we have a 91% viability. So our cells are in the control, live and kicking, and we also have the number of cells per milliliter. So you can easily see what are the numbers, and there comes back the question of always keeping the right volume, both of the solution and of the suspension of your cells, CASY solution and suspension of your cells, because the ratio between both will allow the CASY to immediately give you the values of the number of cells per milliliter. You just have to copy-paste this data. Good. So having done that, we come to the next step, which is now to repeat this procedure. So we need to purge the system, so to make sure that everything is clean and ready and there are no cells left from the control cells. And now we perform or repeat the loading and counting with a different sample. And in this particular case, I've chose the count of a Velkit as an example. And I will stop here for 1 second just to show you, and I hope that you can observe that we now go back to this display of the curve. And you see on the display of the curve again the same values, so the 12 micrometers that defines the distinction border between the cells that are dead and those that are alive. And I hope that you can see now that there's been a number of cells that no longer are within the viable range. So this has dropped considerably. And not only that, we also see here that the viability has dropped to around 70%. And we have the numbers both of the death and of the viable cells. Now, as I mentioned, one of the major advantages of using the CASY is that you have the numbers, but also that you have this graphical display. And the graphical display represents each one of the counts. So you really have a feeling of the Gaussian distribution of the population. And this is important because it gives you a definition of the size. And size, also in cellular terms, matter. The size of a cell matters. Good. Having said that, I come back to the next topic, which is your homework. We are nearly done. Let's assume that I'm showing you this table, and this is the data that you have acquired. So you have the number of cells that you have counted on day 1, on day 2, and on day 3. And you have always the well that has either no treatment or Taxol and Velke treated, and with the two methods, either counting with a CASY or counting with a counting chamber. So you have all of the values. Let's assume that this is the data that you have acquired in your experiment. What I would like you to do is the following. Try to present this data in a graphical form. So to analyze the effect of both inhibitors over time in a time lapse. So an example is shown here in red. This is again not the results of the course, but this is just to show you how you can graphically represent that. So for example, you can perform or represent it in a graph that shows you the time, in this case hours, but you can also put it in the days, the 3 different days, and cells as a percentage. You can either have it as a percentage or in cell numbers. It doesn't matter. But what is important is that this graphical representation will allow you to easily see what happened to the population of cells in the different treatments. So one of them would correspond to your untreated, the other one to the Velcade, and the third one to the Taxol-treated cells. So generate one such graphic and then compare the results obtained with the different treatments. Are they different? Are they the same? What has happened? And then it's also important that you do this not only for the different treatments, but also to the different methods, because you have the results, at least of the viable cells, the cells that are viable, both from the counting chamber and from the CASY counter. So compare the methods. So what are the advantages and limitations of each one of the methods that you have used? And as a last question, if you would repeat this experiment again, and this is a tricky question, what would you change? Or would you make any addition to the protocol? Is there something that we could improve? There is definitely something that could be done, but I hope that you can guess what it is based on the introduction that I gave to you in the beginning of today's session. Remember this table? There is a hint in there. Good. Having said that, I am done with my part, and I would like to remind you of the following, that you will hear from Professor Huber, Lucas Huber, in 5 minutes, how and what you should take as homework and the different steps of data that you should provide for the evaluation. I also would like to ask you a favor, and this favor relates to the evaluation. It's very important for us that you give us feedback. Tell us, how has it been for you to attend this online video practical course, so that we know and we can learn from the process. And this evaluation is particularly important for me because I would like, in the near future, to apply for my Habil. And for this reason, I would kindly ask you to also give me your personal feedback, so you will be asked to give me your comments. Please do so. Anything that you can provide to me will be highly appreciated. And with that, it's all for me, and I leave you. Remember, in 5 minutes, you will have Professor Huber in here explaining you how and what you need to prepare for your homework. All for now. Bye.
A mechanical forerunner of the computer. If you're feeling like a cabbage after too long in the Babbage, and your mind is full of Mendel clams and yaks, then it's time for celebration, 'cause here comes a new vacation. So forget about those lectures and relax. If you're feeling like a zombie after hearing too much Pombi, and your mind is full of yeasts and frogs and flies, you've seen each kinase from the zoo, lots of phosphatases too, and you're seeing cycling cells before your eyes. If you're feeling deep neurosis as he raves about mitosis, with the gleam of microtubules in his eyes, then I think it might be best to try G0 for a rest, and to borrow someone else's college prize. If you're feeling like a cabbage after too long in the Babbage, and your mind is full of Mendel clams and yaks, then it's time for celebration, 'cause here comes a new vacation. So forget about those lectures and relax. If the seats are getting harder and the clock seems to have stopped, and your lower half is slowly getting cramped, then if you try to reach the table where that damn projector's propped, maybe you can reach the wire and fuse the lamp. But if you try to have the last word in a hail of paper planes, so the room becomes an aerial traffic jam, then you might like to remember as you join in all the games, I can always get my own back in exams. If you're feeling like a cabbage after too long in the Babbage, and your mind is full of Mendel clams and yaks, then it's time for celebration, 'cause here comes a new vacation. So forget about those lectures and relax. Go and have your photo taken in the market in Tangiers, or maybe go and see the sights of Greece. But the only sights you'll see are 100 other students, and you saw all them the year before in Nice. As for me, I'll stay at home, no longer will I roam. I don't care what the staff and students say. I'll buy a sunray lamp and a set of foreign stamps, send myself a reprint request every day. 'Cause I'm feeling like a cabbage after too long in the Babbage, and my mind is full of Mendel clams and yaks. But it's time for celebration, 'cause here comes a new vacation. So forget about those lectures and relax.
Ja, liebe Kolleginnen, liebe Kollegen, dear colleagues, now we come to the summary and the most important part for you, describing the homework. This was a big experiment for all of us. It was an experiment for you guys at home, watching a practical course online, something I could never have experienced before myself. But it was a challenge for us to provide you with something we hope that it would be useful for you. We took many efforts, and it took us quite some time to record these videos for you and to play all the experiments through, but we learned a lot. And what we learned, for instance, was that with those video tutorials, we are able to explain many things much better than in the real practicum room itself, because you are 20, 30 students around. the many details how to take off a supernatant without disturbing or floating up the pellet, how to load a sample properly on a gel, all these things, all these little details became much more visible due to these efforts we did with the video streaming. Now, of course, we are not living in a perfect world at the moment, and we all suffer from the restrictions from Corona. And however, we need to use the time provided, and we all need to put our efforts into having good distance teaching, and for you that you learn something and you can take something with you. Therefore, we have you supplied with video material, with PDF files on the Elias, and I will go a bit for the material that we have supplied for you for the homework. In the worst case scenario, this was the practical course, because I basically and honestly have no idea if we will be able to teach you in September. I have tried to move this practical week very far back in September in the hope that the situation might clear up considerably, but we don't know. If things go wrong, that means we cannot have a practical part really in the lab, then you at least have had a practical introduction with some videos that you can imagine what the work would be about. And based on that, what we have presented to you, you can do some homework that we can evaluate. You can do the homework alone, you can do the homework in teams, but everybody of you has to deliver something like a protocol. And this protocol will contain answers to questions that we asked you during the course, the different tutors, that I will summarize again for you, that you also have in Elias as a PDF file, and it will also contain some other work you need to do. In the case that things develop pretty well during that summer and we are able to do some practical work, you are trained well enough now to move in small groups into the practical room and to perform some tissue culture work, to perform some gel electrophoresis, or even a subcellular fractionation or something along those lines. And we are absolutely willing to provide you with that, but this does not depend necessarily on us. I am also giving you this homework description and the final summary of the practical course in English for two reasons. First reason is that we are teaching this course in English in order to train you. And secondly, more and more students from universities abroad, so from Ukraine to Germany and to Italy, have asked if they can get access to these practical courses and to the lectures on our channel and on our stream. And therefore, I will also describe this homework in English for this reason. Now, let's go to the homework now. There are questions that we kindly ask you to answer in the next slides. Then you should write an essay. An essay is a short scientific description on a certain topic in your own words. And this essay should be about subcellular fractionation. And that is based on experiment number 2 that was introduced to you by Tara Stasik. And this was the online stream on May 13 and 14. And we have provided you, in order to make it a bit simpler, a literature for that. And that is a book chapter that we wrote last year about subcellular fractionation in order to provide a guidance for scientists out there how they can use it. This is this book chapter that I have mentioned. It was written by Mariana, and it's about subcellular fractionation, and it contains pictures that you have seen during the course of this practical stream. It contains passages of protocols that you have seen in the protocol that we gave to you. And it gives you a bit of a framework about the methodology that we are introducing to you, about its limitations, and it's about potential applications. I come to that point back at the end of my introduction. Homework. This was the question that Mariana has raised on day one. She showed you 3 videos, AVIO MP4 files that have been uploaded, and she asked you questions to get the answers. This was which of the videos that we explained during the course is the untreated, the Taxol treated, or the well-cat treated. The videos have been uploaded for you again, that you can look at them at higher resolution than it would be possible, for instance, in the stream, in the online stream. Look at them carefully, compare it to what you have heard in the stream from Mariana, and write a little answer and justify your answer. Why you think video C corresponds to treatment X, Y, or Z. These are questions that are raised by Ilya Vietor, who introduced you to the cell passaging, to the HeLa cell culture protocol, and to immunofluorescence. And some of the questions you have been asked already during the stream, some are new. So for instance, what is the function of the phenol red in the culture medium? Which 2 proteins were transfected actually in our experiments? Where, at which organelles, so to say, should they localize? How do we detect them in the cells? Which organelle should be LAMP1 marker positive one? What co-localizes with LAMP1? So a question along the same lines in immunofluorescence, RAP5 or RAP7. How do you detect nuclei in immunofluorescence microscopy? And the last question, what do we mean under negative control in the immunofluorescence detection? So you write down in the protocol these questions and you write your answers right underneath. So we are filling this protocol. Now comes a question for your homework concerning experiment 2, the subcellular fractionation, where we needed to establish the amounts of proteins that we are going to use for the gel electrophoresis and the Western blotting afterwards. And these were the results from the Bradford assay. So you have the ODs that we read out, and here's the question that you need to calculate the volume that we would load up on the gel. Next question, result from the Western blotting that we virtually performed altogether. Analyze these Western blots. Questions are, find out the order of the fractions that are loaded. Taras has introduced to you that each student, student 1, student 2, and student 3 has loaded 3 samples. But we have different antibodies used. So find out the order of the fractions, draw conclusions on the distribution of the markers between the different subcellular fractions, write these questions up and fill the answers right underneath. Now comes a question to experiment number 3. We have done the cytotoxicity assay, the WST1 assay. And here you get a graph from the experiment, a Taxol and well-cat treated sample. And it's a quantitative measure. It shows you how much substance is needed to inhibit a given biological process by 50%. This is the IC50 and this is cell growth. So now calculate these IC50s for the different treatments. This has been explained in the streams. Now we come to experiment number 4, which was introduced today by Mariana to you. The questions are as follows. Which of the methods from the introduction table, you remember this big table with the different methods, would you use to count A, T cells isolated from the spleen of a mouse, or B, proliferating cells on a skin biopsy? Justify your answer. So these are 2 completely different samples. The question is the same. You want to know how many cells proliferate in there. but there are different samples, therefore you need to apply different methods, and therefore you have to revisit Mariana's stream and look in the table and listen carefully what she has explained, and also look into the literature. Then a question where you need to Google a bit. The Neubauer counting chamber that was introduced to you has a different name historically. Do you know anything about it? Would you find something out about it? Write this question and write the answer right underneath into your protocol. Another homework from experiment 4, today the proliferation part, was introduced in the stream to you. Let's assume this table bill here represents the number of cells you have counted today from the different time points of the treatment using the CASY counter or the counting chamber, it does not really matter. And you have different number of cells. You have ones with V, this corresponds to the number of viable cells, V like viable, or D like dead to the number of dead cells or death, cell death. And the question that arises from that are 5 sub-questions. You should analyze graphically the effect of the inhibitors over time. Therefore, you consult Mariana's stream again. Compare the results obtained with the different treatments. Compare the results obtained with both methods. And what are the advantages and limitations of the chosen methods? If you would repeat the experiment again, would you change something in the protocol? You write that questions in your protocol and the answers right underneath. Finally, the part that is a bit challenging for you, and this is to write an essay. This does not need to be very long. It should be 3 to 4 pages at the maximum. It should be in English. Use your own words. We gave you some literature, a PDF file of an article describing subcellular fractionation. The topic of your essay is subcellular fractionation. Write what the method is about, why is it useful, what you don't find particularly useful or interesting, what you could do better, or what you find good, how you would interpret it, what are the limitations. You can accompany that with online material that you find with your own searches or what we have uploaded for you. The rules are easy. Use your own words, your own English. I will, when I correct that, recognize immediately when you use cut, copy, paste from other examples of the literature. I'm correcting and reading many bachelor theses, master theses, PhD theses, scientific papers, reviews. I can judge the level of your English, so don't try to fool me. Write a little article by yourself. You learn about the English, about the scientific English. I will correct it, I will give you feedback. Don't be afraid, use your own words. Use figures, you can draw your own figures, you can draw them with your hand. You all remember the seminar you had in your bachelor studies where I always encouraged you maybe to draw your own slides with your own hands to find your own material. When you use material out of the literature, cite it properly, write a little introduction, give the entire thing a title with your name and affiliation with your ID numbers from the university, then write the introduction and write the main part and maybe a conclusion summary part, and then cite the literature at the end that you would like to cite. And when you insert graphics, give a legend to the graphic, explain what is it about, a picture, and when you cite something from another scientific literature, from another website, cite it properly. All that, the entire package of your homework, you have to send back until Friday, May 29. This is exactly 2 weeks from now, to my email address, which is given here below. In case you are an external student from another university who is also joining our course, you are also welcome to participate in this exercise, and I take it as my obligation then also to help you and to supervise you and to give you some feedback. So now, my dear students, this is the end of this exercise. It's not a perfect world. we did our best. We tried it. We had fun doing it, actually. It was very exciting for us. I hope you enjoyed it a bit. And I hope it helped you getting through this difficult time by learning something new, something unexpected, and giving you a bit of appetite what will come in your life now as bachelor, later master students or PhD students. When you come to our labs, do your exercises there, maybe do your thesis with us. It's an exciting scientific world outside there that is waiting for you. And with these words, I say goodbye to you guys. I remain in the stream online. If you have comments or questions, I will kick out my picture now and the presentation. But I will remain online, and you can ask me questions, or you can send them by email to me or to my co-workers, and we will be happy to answer them. And have a good time and stay healthy.
Yeah. Corona 2020 recordings. Corona 2020, ein richtiges Luder, aber macht die Rechnung nicht ohne Hest, Heiß und Huber. Online-Vorlesung geht ab, ziemlich nice. Um 11 Uhr stehen wir alle bereit, transportiert wird der Vibe ohne Spannungsgradienten. Huber, Hess und Heiß, die Unwissenheit wird enden. Sag mir, wer liefert ab? Heißer als ein Trio, das Zellbiologie-Trio.
Wir sitzen alle da, sind am Ende mit unserem Land. Es kann doch alles nicht sein, aber wir sind niemals allein.
Come on everybody!
sind am Ende mit unserem Latein. Das kann doch alles nicht sein. Aber wir sind niemals allein.